Hello, everyone. Today we will be discussing the effect of different types of radiation on the biological processes of Daphnia magna, a keystone consumer and prey animal in aquatic ecosystems.
The focus of this experiment was to understand the impact of induced forms of radiation on Daphnia magna and how it can affect the population, fertility rate, and survival of the species. This research is significant because it sheds light on the consequences of radiation from anthropogenic sources such as cell towers, microwaves, and excess UV radiation passing through holes in the ozone on public health, animal populations, and the environment.
To perform the experiment, five types of radiation were tested on Daphnia magna, including ultraviolet radiation, visible light, radio waves, microwaves, and a control group. The Daphnia were exposed to each type of radiation for 3 minutes every 9 minutes for 18 minutes, and the population count, vertical distribution, heart rate, and death rate were recorded.
Here are the pictures for the experiment.
The results of the study on the effect of different types of radiation on Daphnia showed that exposure to UV radiation had the greatest negative effect on the egg count. However, results regarding the fertility rate of the Daphnia proved to be inconclusive as no new Daphnia magna had hatched during the experiment. In fact, all Daphnia lost their eggs by day 10, aligning with the natural 8-day egg cycle. A possible reason as to why this occurred could be the expulsion of resting eggs from the females as opposed to live young, which could have resulted from unsuitable conditions within the test tube either due to radiation or depletion of oxygen over time.
The average heart rate of all the groups increased over time, which indicates increased stress levels and difficulty adapting to the new environment. However, the heart rate of the control group had the highest simple average regression slope, as seen in the statistics table. This was unexpected as the other dependent variables of the experiment showed the most negative results towards the UV group, making heart rate an outlier.
The data for the diurnal vertical migration, a behavior used by Daphnia to protect themselves from radiation, showed that the UV radiation group started off lower and became even lower, indicating a reduced ability to protect themselves. If diurnal vertical migration becomes too pronounced, more frequent predator prey interactions occur, with Daphnia being the prey, destabilizing aquatic food chains where Daphnia are found.
The Daphne exposed to UV radiation expired by day 10. The population had a standard deviation condensed around the mean, showing a correlation between radiation energy and higher death rates among Daphnia within the test tube. The Daphne exposed to microwaves and UV radiation followed a similar pattern, with all test specimens dying by day 10. On the other hand, control, visible light, and radio wave groups were more similar to each other, dying at day 14. This resulted in the experiment's termination.
This experiment was conducted to gain insight on how radiation can affect animal populations. Unfortunately, the experiment was terminated by day 14 due to the death of all test specimens. However, enough data was collected to make a conclusion. Overall, the results supported the hypothesis, made evident by the fact that the group exposed to UV radiation had the worst overall health in 3 out of the 4 categories. The results of this experiment will provide us with a better understanding of the impact of radiation on Daphnia magna and the ecosystem. This information can also give us insight into how other organisms, including humans, can be affected by radiation exposure.
An article written by a team of researchers led by Lars Anders Hansen measured photoprotective compounds showing more of a thorough understanding as to how Daphnia have mechanisms protecting themselves. The diurnal vertical migration showed a behavioral adaptation, but a biological one, more specifically hormonal, chemical, or physical, would provide additional insight. However, this would require professional lab equipment, thus would demand a higher budget. Earthworms could be a better test specimen as it eliminates the need for water, which may have caused inconsistencies. Finally, more radiation types could be used, such as infrared or gamma radiation, to test a wider range of the electromagnetic spectrum.
This is the bibliography used for the experiment. Thank you for listening.